for us right now, we're going to be looking at whether or not this chemical, which is related to amyloid beta, related to Alzheimer's, we're going to be putting it into this model of dementia, this mouse model, and in stem cells, and see if we can inhibit the formation of dementia. We're also going to use it in diabetic animals and see if we can stop the diabetes from being a risk factor for the Alzheimer's. So right now we're looking very much at being able to use a treatment directly into the brain to be able to inhibit the development of the dementia. If you show it works in mice, you show that it's safe, then it goes on to preclinical trials where you look at other animals, maybe pigs, monkeys, something like that. The most important thing is you show that it's not a harmful treatment. In a lot of diseases where the disease is not always fatal and it's not always serious, you will not be allowed to do a treatment that could be very radical, very revolutionary, because it's so dangerous. In diseases such as, for example, pancreatic cancer, which has a survival rate that's very low, or Alzheimer's, which has a survival rate of 0, you are allowed to do treatments that might be more revolutionary, more high risk, because what's the downside, right? And a lot of the people who have these diseases will say, we could say, well, this treatment may kill you. Well, you have a disease like pancreatic cancer, something like that, that's going to kill you. It would probably first be looked at by just putting the chemical into the brain of these people or into their bodies, see if it had any positive effect. If it does, there's no downside, then it could go into stem cell treatment. I mean, stem cell treatment is really the horizon that people are looking at. Being able to either put in a stem cell that makes something you want, or to be able to put a virus in that transmits the gene you need directly to the cells that are there. So those two approaches are two that people are really looking at for curing a disease. If a person pancreas doesn't make insulin, maybe you can put viruses in there that take the insulin-making gene and put them into the cells. The cells will start making insulin, and you've cured the disease. If the brain is becoming demented because it doesn't have a properly functioning insulin system, maybe if you put stem cells in there, that they become neurons, and they will pump out this chemical that will replace the insulin. Maybe you've cured the disease. So we're looking at things where you're going to where the body will make the drug in a controlled way, and not a drug that you're taking externally. So the nice thing about if you were going to use stem cells, theoretically, what we would do is we would take some tissue from you, probably skin, revert the cell to a stem cell. And once it's a stem cell, it divides like crazy. You get all kinds of test tubes full of your own stem cells. Then you do whatever genetic manipulation you want to the stem cells, put it back into your body. It's your own cells. So we don't have to take stem cells out of embryos anymore. And I think that was the controversy. And the stem cells we're talking about aren't what you would call pluripotent, which means they can't just become any kind of cell. The stem cells we make are neuronal precursors, so they become neurons and cells that are like neurons. So it's much easier to form these, and you can form that from skin. But you can make real stem cells out of people's own tissue, and they seem to be able to do just about anything that we would ever want a stem cell to do. So I think a lot of the controversy originally was the embryonic origins of the stem cells we don't have right now. Now, what some people are doing, which is really brilliant, is they are taking some cord tissue, so from the amniotic cord, taking those cells and freezing them away in case sometime when they're an adult, you have real hardcore embryonic stem cells from when you were an embryo, right? But I think within a few years we'll be able to do that easily from our own cells.